The next talk is going to be recorded video by Insub Kim from Stanford University. However, Kalanitz Grill-Spector will be here to answer questions, so please stick around.
Yeah. So sorry, Insub could not come because of visa issues. So he's pre-recorded the talk, and then I'll be here at the end to answer any questions. So how do we start playing?
Hello, my name is Insub, and sorry I could not be there in person. It's a bummer, but here's my talk. Retinotopic maps, which characterize the relationship between the field of view of the external world and the brain, has been extensively characterized across multiple labs and the individual level. To obtain these retinotopic maps, we typically use stimulus like rotating wedges, expanding rings, and moving bars. These stimulus are used to stimulate different parts of the visual field one by one. We use population receptive field model to estimate the region in the visual field that is processed by the population of neurons in a voxel. Additionally, there are category-selective maps in the ventral temporal cortex, which are regions of the brain that respond strongly to a handful of categories than other visual stimuli. To map these regions, stimul sets are designed to include images of faces, bodies, places, objects, and words. By contrasting the fMRI responses elicited by one set of category to the rest, we can obtain category-selective visual regions. In this example, we are showing a CoS-places area by contrasting places to all other categories. So there are two different experiments because each experiment is targeting two different visual properties along the visual hierarchy. And the goal of this project is to create a new set of optimal stimuli that integrates distinct experiments into one, so that we can map retinotopic and category-selective regions within a single experiment. In this new record experiment, we use naturalistic images of 5 different categories, such as faces, bodies, places, words, and food, due to its recent discoveries. To do so, the record experiment needed to elicit distinct responses in different population receptive fields and category-selective regions. So how did we do this? We needed a systematic way of generating images. We used a pre-trained stable diffusion model to generate stimuli for the record experiment. We chose the stable diffusion model because previous research showed successful reconstruction of what people visually saw from fMRI data. So here is a typical diffusion model where the model takes noise and text prompts as its inputs. For the text prompts, we crafted descriptions to match the representation of 5 different categories. For example, for food, we typed bibimbap, rice, spinach, a fried egg. So given these inputs, the denoising unit iteratively generates images, and a decoder converts the latent representation back into an image. As you can see, the generated image looks naturalistic and matches the text prompt descriptions. And this is to elicit distinctive responses in category-selective regions of the brain. To optimize for retinotopic mapping, we simulated 100 pRFs that mimicked the characteristics of V1, and to predict fMRI time courses from these pRFs. We first computed the local contrast of images because that is what drives V1. Then predicted fMRI time courses from 100 pRFs tiling 12-degree field of view, and their size increasing with eccentricity. The generated image sequences will be considered optimal if they induce distinctive fMRI time courses for each of the 100 pRFs. Leveraging the image computable nature of the pRF model, we iteratively refined the image generation procedure using classifier-free guidance. In this approach, we iteratively perturb the image to minimize the covariance of 100 predicted fMRI time courses, ensuring distinctive responses for different pRFs. It is important to note that the temporal sequence of images is crucial because both predicted fMRI time courses and image generation process are influenced by the order of image presentation. Here, I'm showing one example run of the fMRI experiment, which has a sequence of 70 images containing 5 categories with 14 unique images per category, and each image was presented at a rate of 1 Hz over a 4-second trial. With this new record experiment stimulus set in hand, we conducted 6 runs of the experiment. While viewing the images within the scanner, participants were instructed to press the button whenever they saw a target color in the fixation. As a comparison, we conducted 4 runs of pRF experiment with moving bars and 4 runs of functional category localizer with food. This is the retinotopic maps estimated from the record experiment. Surprisingly, even with naturalistic images, we could successfully obtain polar angle, eccentricity, and pRF size map. If we compare them to the maps of the pRF experiments, we can see very similar maps between the 2 experiments. While the maps obtained from the pRF experiment covers more of the ventral regions beyond V4 and VO. When we quantitatively compare the pRF parameters from an example subject V1, the estimations of pRF locations X and Y are very similar to each other, as they are on the diagonal. For the pRF size, we can replicate well-documented pRF size versus eccentricity relationship, where the pRF size increases with eccentricity. And the estimated slope for each of the experiment were very similar to each other. But if you look closely, there were some differences. The pRF experiment inaccurately estimates many voxels to have sizes near 0, while the record experiment showed more size variability towards periphery. Now, let's look at the category-selective maps across the 2 experiments. When we contrast image of faces with other stimulus and threshold it by the t-value of 3, we get activation along the fusiform, as expected. And the activity pattern is very similar across the 2 experiments, as you can see here. By applying the same procedure to all other categories and tracing their selectivity pattern based on anatomical landmarks, we can obtain very similar category-selective ROIs across 2 experiments. Finally, here we are plotting category selectivity for each category by taking an anatomical ROI of right hemisphere ventral temporal cortex. As shown here, most of the dots are on the diagonal, showing high correlation between the 2 experiments at the individual voxel level. However, we see weaker correlation for the word category, potentially due to having overall weaker signal or selectivity in both experiments. Using a generative neural network framework to generate sequence of images, we simultaneously obtained retinotopic maps and category-selective maps. We demonstrated the potential of generative neural networks for designing optimal and efficient experiments. Right now, the differences are 32 minutes versus 28 minutes, but we are improving it. And another benefit is that you can generate endless of images based on the prompt. And this framework is applicable to any image computable model. For example, if a model computes how memorable an image is, we can generate images to maximize memorability using the same framework. With that, I would like to thank my collaborator, Jen, and the VPNL lab. And Colin will be there to answer all your questions. Thank you.
Thank you very much. So if you have any questions, you can come up to the microphones, and we'll start with the question from the audience on the Q&A. Were word stimuli real words rather than DNN hallucinated non-words?
All the word stimuli in the experiment are DNN hallucinated words. Sometimes they make sense and sometimes they don't make sense at all.
So not all real. Equally, there is another question. Some stimuli look not very natural. What that affect? Will that affect the results?
So a lot of these high-level visual areas responds to abstract stimuli, so line drawings or very impoverished stimuli also activates these regions. We haven't looked at an item-based level yet to actually see how natural looking it is, how it affects the response per each category, but we can do this because we have individual stimuli responses.
Okay, thank you very much. There aren't any questions from the audience. So
And if you have any more questions, come see the poster and I'll be there. Thank you very much.
If you can, please fill in the survey to give us feedback about this session. It's just through the app. Thank you.